The speaker in the movie will be Yael, and behind the scene, Ruth Sorek Abramovich, who might be able to answer a couple of questions at the end.
Yes, I'm here, ready.
The title is Temporal and Spatial Analysis of Forward and Backward Microbial Contaminations in a Mars Analog Mission.
Hello, my name is Yael, and today I'm going to present my research titled Analysis of Forward and Backward Microbial Contamination in a Mars Analog Mission. I guess at least some of you watched one or more of these sci-fi movies, and in case that you didn't, I'll tell you that all of them revolve around one main plotline: bringing an alien life form from extraterrestrial ground and risking the human race. After I'm done with this presentation, you'll know that it's actually the dreaded case of backward contamination. So what are forward and backward contamination exactly? Let's look at Mars as an example. Forward contamination is defined as the possible transport of Earth-based microbes to Mars or to other celestial bodies, while backward contamination is defined as the possibility that extraterrestrial microbial life returned by a space mission can be established and proliferate on Earth. Backward contamination is less likely to happen, but if it does, it can have severe implications. Protection against contamination has significant ethical, scientific, and safety-related aspects. Forward and backward contamination is a risk that cannot be ignored, particularly due to the increasing number of planned spaceflight missions. Ethical aspects of this topic states that the extraterrestrial environment must be protected from any biocontamination by terrestrial microorganisms, in order, of course, to avoid any harmful interference with either putative local life or the environmental conditions. Scientific aspects states that the exobiological experimentation results must be protected in order to avoid any misinterpretation that would lead to a false discovery of traces of life in an extraterrestrial sample. Another option is the non-discovery of extraterrestrial life on a sample because of its degradation as a result of poor preservation. Safety aspects states that the Earth biosphere and also the moons must be protected against possible contamination, which could be included as returned samples or carried by probes or crews. A recent example of forward contamination risk is the case of the Israeli space Yael Bereshit lunar lander that I guess that all of you heard about. Bereshit's hard landing on the moon in April 2020, while carrying a small payload of tardigrades, which are also known as water bears, raised an intense global debate on both ethical and legal implication of a possible forward contamination event. One way for us to study contamination issues and other space-related technological and scientific questions is by simulating analog missions. Analog missions are field tests in locations that have physical similarities to the extreme space environment, such as isolation, radiation, geomorphology, and more. Obviously, no analog can simulate all aspects of human space missions here on Earth, and this is why a wide array of analog activities are necessary. Ideas and technologies can be tested in analogs before trying them in space. Those that do not work in analog mission will not be launched. Ground-based analog studies are completed much more quickly and less expensively, and regarding contamination issues, several in-depth analyses of contamination potentials were previously done in a Mars analog mission. For example, the AstroMars simulation that was done in MDRS Center in the Mojave Desert. Here you can see a collection of Mars and lunar analogs on Earth. For example, you can see the Mars 500 project in Russia and NASA's NEMO underwater station in the US, and of course many others. Our aim was to utilize, for the first time to our knowledge, advanced molecular microbiology tools to better understand cross-contamination potential at a setting of an analog mission. We hypothesized that throughout the mission, if forward contamination will occur, environmental microbial population diversity and composition will be affected by the artificial introduction of new taxa, potentially human-associated species. If backward contamination will occur, microbial species from the environment will be carried on the astronaut's glove and will be introduced inside a habitat. So as I said, we aim to investigate the backward and forward contamination potential in a Mars analog. We chose the Ramon crater for an analog mission due to its Mars-like geomorphology, geology, isolation, and hyper-arid climate conditions. This study was done in collaboration with Desert Mars Analog Ramon Station, or in short, DMARS, the first Israeli Mars Analog Ramon Center. Mars Analog Center. Between the 3rd and the 14th of March 2019, an 11-day analog mission took place. During that mission, 6 analog astronauts, with me as one of them, performed daily extravehicular activities while fully suited with a simulated spacesuit and a personal life support system. So we will start with the forward contamination part. A crucial step in the study design was the site selection. We chose 3 sites. 2 of them were frequently visited by the astronauts, and the other one was an isolated sterile site. The first site was the entrance site, located around half a meter from the habitat's airlock, which means that upon each leave and return of the astronauts to the habitat, this site was interrupted. The sanitary site was located at the back of the habitat, about 30 meters from the sanitary disposal tank. This site was also frequently visited, although less than the entrance. The far site was approximately 280 meters west of the habitat and was banned for entry except for sampling. This is an orthophoto and elevation model of the mission area and sampling sites. You can see the relative location of the far site compared to the habitat and sites S, which means sanitary, and A, which stands for entrance site. To study forward contamination, we sampled our 3 sites on the 3rd, 5th, 7th, and 9th days of the mission in duplicates. On each sampling day, the DNA from the soil sample was extracted inside the habitat using a biological hood to minimize contamination. After the mission, we performed microbial population profiling using PCR amplification and sequencing. Due to the shortage of time, I will only present the results for the 16S analysis, which are representative also for the 18S. This process allows us to determine the taxonomical identity of the microbial and archaeal population of each site. This is how a sample collection during EVA looks like. Specifically, those are pictures of the sanitary site and the far site. Moving on to the results. As I mentioned, we sampled each site on 4 different time points during the mission on duplicates, which means a total of 24 samples. Each sample was analyzed to determine the population composition and the dominant taxa. Here you can see a principal coordinate analysis, or in short, PCOA. Each point of this graph represents the population composition of one sample from one site from one time point. The closer 2 points are, it means that the more similar the population composition is, and the further they are from another, their population is more different. As you can see, we observed a strong separation between the 3 sites, with samples collected from the far side in the circles have very similar composition. In order to better understand the population dynamics, we closely examined the microbial taxa found in each sample. Each point you saw on the PCOA from the last slide is now 1 bar. Here you can see bar graph representing the relative abundance of bacterial families in the samples throughout the mission. Each color on the graph represents a distinct family, and close color represent close bacterial families from the same order. Only taxa that constitute over 1% of an individual sample are presented, meaning that taxa that were found in very low amounts are left out to simplify the visualization. The numbers in the sample name represent the sampling date. 03 means samples taken on the 3rd date, and so on. The entrance site that you can see on the right was the most diverse site of all 3, with rapidly changing composition, which align with it being the most visited site. Some taxa were rather stable in all time points. and the far side that you can see here on the left had relatively high and stable diversity across all time points. The sanitary site was the least diverse site, with particularly low diversity observed for some samples taken on the 7th and 9th day, something that we saw also in the entrance site. Moraxelocea was the dominant family across all sites, with the genus Psychobacter most prevalent. This environment seems highly disrupted, especially when compared to the stable and diverse far site that you can see here. When we search for forward contamination events, we search for human-associated microbes that appear somewhere that they should not be. Human-associated microbes doesn't necessarily mean that they are pathogenic. For example, they can be part of the human microbiome. To identify these bacteria, we first compiled the list of taxa found to be human-associated in previous studies. To do that, we used the dbBact database. dbBact is a community knowledge base for information about microbial ecology. The human-associated taxa that we found can serve us as the markers to detect forward contamination. Here you can see the relative abundance of human-associated genera in the samples taken from each site. The sanitary site presented the highest relative abundance of human-associated genera, with samples collected on the 7th day being the most contaminated. As an example, the genus Citrobacter had an average relative abundance of 0.09% on the 3rd and the 5th day, and on the 7th day, its relative abundance in samples taken from the sanitary site increased to 38%. The entrance site was less contaminated, but still had higher relative abundance of human-associated genera as compared to the far site, which presented very low to non-existent level of contamination. To summarize this part, we were able to detect possible forward contamination events in which human-associated genera were found in 2 sites, especially at the sanitary site. Moving on to the last part of my talk. In order to study backward contamination, we designed the following protocol. Nitrile gloves were placed over the astronaut suit glove in the airlock right before leaving to an EVA. Section of the palm of the hand and then the 5 fingers were gently swabbed using a sterile cotton swab. The second sampling took place immediately upon the astronaut returned back to the habitat. Both samples were extracted and treated as I previously described. We analyzed the glove samples in a similar way to the soil samples to study the population composition of each sample. Before samples refer to samples taken before leaving the habitat, and after samples were taken after the EVA in the simulated airlock. The principal coordinate analysis indicated that the before samples clustered together along with the controls, suggesting a similar population composition. In contrast, the after samples were much more dispersed and did not cluster with the controls. As before, we took a closer look on the dominant taxa found in each sample to detect microorganisms carried on the astronauts' gloves upon return from EVAs. Baseline for the nitrile gloves was conducted, so each after EVA sample was compared and paired to its before EVA sample. Each 2 bars represent 1 astronaut, and samples were taken on the 3rd, 5th, and 7th day. What we saw here is a different microbial composition between the before sample and the after samples, indicating that microbes were indeed carried upon the gloves. To detect the most probable candidate for backward contamination, genera detected from after EVA gloves were cross-referenced with those detected in the soil samples. After we did that intersection, we got 44 genera that were shared between both samples type, meaning that 44 genera were carried on from the environment into the habitat. We then examined the distribution patterns of these shared genera across the soil site to detect the most probable markers for backward contamination. 10 shared genera out of the 44 I mentioned appeared mainly in the far site and belong to a commonly arid soil taxa such as Chlorophlaxia, which is a green non-sulfur bacteria, and Solirubobacter. These may be viewed as classic examples of backward contamination. An interesting case is of the member of the Moraxelocea family, the genus Psychrobacter. This one presented a possible case of a cross-contamination event, which means it was taken from inside the habitat to the outside, and then back again inside the habitat. This genus was found with low relative abundance in the before samples taken on the third day, and upon return to the habitat from an EVA, it was found in much higher relative abundance. Psychrobacter was present in sites F and A, far and entrance, with extremely low average relative abundance, and showed high relative abundance in the sanitary site, suggesting its source is not the local environment. We verified that what we see is not an environmental strain of Psychrobacter, and indeed we found that it's most likely to be human-associated. To summarize, using amplicon sequencing, we investigated the possible occurrence of forward and backward contamination during a Mars analog mission. We show that forward contamination occurs in the area in close proximity to the habitat. We also show that backward contamination occurs as we detected environmental bacteria that were carried upon the astronauts' gloves. It is possible that the presence of Psychrobacter in the sanitary site is due to forward contamination, and its return to the habitat is an evidence of backward contamination, hence cross-contamination. We show that analog missions are in established settings to study contamination questions. To conclude, the ability to control and reduce forward and backward contamination is crucial for the future of humankind in space. I want to thank all the interdisciplinary members of this study. As you can see, it's quite across centers and universities. And of course, the funding that was given to us by the Israeli Space Agency and the ICA Foundation. And thank you very much for listening. If you have any questions, please free to ask my colleague Reut or email me. Thank you very much.
Okay, any questions with you?
So there's one question that it appears that human exploration implies concomitant forward contamination. Realistically, would it be feasible to prevent it completely each time, for example, during space exploration?
So I think the practical answer for that is no. You cannot completely avoid whatever we bring with us. And also NASA and the American National Academy of Sciences has decided several decades ago that there's no need to fully sterilize all the rovers and spaceships that NASA has been sending to outer space and has been sending to Mars, for instance, because we are not really good at complete sterilization of anything. So there is a big workshops and big discussions going on with the with COSPA and planetary protection committees and NASA people to at least understand what are the gaps in our contamination knowledge. And this work is one type of an answer to those kind of gaps that we have in our contamination knowledge. So there's a lot of issue to how to avoid it, how to monitor it. This work is more about a suggestion on how to monitor such an event. And I think it's a much more practical way than to say, okay, let's sterilize everything again and again and again. It's not practical. It takes a lot of time and energy. So that's where we're at the moment.